welcome to the Deep Dive. Today we're asking a really fundamental question, maybe the fundamental question in biology. What actually makes something alive? As opposed to just, a rock or a machine?
It's a huge question. And the answer, well, it really comes down to one thing: the cell. It's that basic unit, the foundation for absolutely everything living.
Exactly. So that's our mission for this deep dive, to really get into the cell. We'll look at how it was discovered, which is a great story in itself, the theories built around it, and then really unpack all the amazing bits inside and what they do. How do these tiny parts work together to make life happen?
And we'll be looking at this through a lens. Scientists often use something called reductionist biology. It sounds fancy, but it's basically a physicochemical approach.
Meaning?
Meaning we use ideas from physics and chemistry to understand biology right down at the molecular level. Figure out what individual molecules are doing, how they build structures, how they carry out functions. It even helps us understand what goes wrong in diseases.
Right, getting down to the nitty-gritty molecules. And speaking of that deep level, we should definitely mention G.N. Ramachandran, an absolute giant in protein structure.
Oh, definitely. The Madras school he founded, hugely influential. His work on collagen, that triple helix structure, groundbreaking.
And the Ramachandran plot. Anyone who studies proteins knows that plot. It basically maps out which shapes protein chains can stably fold into. It just shows the incredible detail we can now understand about life's building blocks.
It's a perfect example of that reductionist power, yeah. Seeing the rules governing the molecules of life.
So how did we even start seeing these tiny things? Cells aren't exactly visible to the naked eye.
No, not at all. It really hinged on technology, microscopes. Their invention and then constant improvement was absolutely key. You had Antony von Leeuwenhoek way back, who was the first person to actually see and describe a living cell. Amazing moment.
Just peering into this unseen world.
Exactly. And then later, Robert Brown, he identified the nucleus inside the cell, that sort of central hub. But it took time for these observations to come together.
Which leads us to the big idea. Cell theory. How did that develop?
Well, it started gaining real traction in the late 1830s. You had Matthias Schleiden, a German botanist, in 1838. He looked at tons of plants and realized, hang on, all these plants are made of cells. Different types, sure, but always cells forming tissues.
Okay, so Schleiden's focused on plants.
Right. And then, almost at the exact same time, 1839, you have Theodor Schmann, a zoologist. He's looking at animal cells. He noticed that thin outer layer, what we now call the plasma membrane.
The boundary.
And he compared animal and plant cells and realized the cell wall, that rigid outer layer, was unique to plants. That was a key distinction.
So putting their work together.
Schleiden and Schwann proposed this hypothesis, basically that animals and plants are all fundamentally made of cells and the stuff cells produce. A huge conceptual leap.
But there was a piece missing, wasn't there?
There was, a big one. Their theory was great, but it didn't explain, well, where do new cells come from? Did they just pop into existence?
Right, the origin question.
And that was answered by Rudolf Virchow in 1855. He made the crucial observation and statement, cells divide. New cells only come from other, pre-existing cells. Omnis cellulae cella. Every cell from a cell.
Omnis cellulae cella. Got it. So that finalized the core ideas.
Pretty much. Modern cell theory really rests on two main points now. One, all living organisms are made of cells and their products. And two, all cells arise from pre-existing cells.
Simple but profound.
And it highlighted something else too, something quite deep. You only get the phenomena we call life metabolism, response, reproduction, all of it when you have that intact cellular organization. Break the cell apart and those living properties disappear. The whole is definitely more than the sum of its parts.
Okay, so that's the foundation. Now, when we talk about cells today, we usually start with a major split, right? Prokaryotes and eukaryotes.
That's the fundamental divide, yes. And the main difference boils down to the nucleus. Eukaryotic cells, think plants, animals, fungi, protistus, they have a true nucleus. Their genetic material, their DNA, is enclosed within a membrane-bound sac.
In
prokaryotes, bacteria, archaea, they lack that membrane-bound nucleus. Their DNA is just located in a region of the cytoplasm, but not enclosed.
But both types have that internal stuff, the cytoplasm.
Absolutely. The cytoplasm is like the main arena inside the cell. It's this semi-fluid matrix filling the volume, and it's where most of the chemical reactions happen, the stuff that keeps the cell alive and functioning. It's common to both prokaryotes and eukaryotes.
Now, you mentioned diversity earlier. Just how diverse are cells?
Well, incredibly diverse. It's really quite amazing. In size, for instance, you have mycoplasmas, which are probably the smallest cells known, maybe only 0.3 micrometers across. just unbelievably tiny.
Wow.
Then bacteria are typically a bit larger, maybe 3 to 5 micrometers. Compare that to, say, a human red blood cell, which is about 7 micrometers.
Okay, still microscopic.
Definitely.
Yeah.
But then you have the other extreme. The largest single cell is actually an ostrich egg. And think about nerve cells in your own body. They can be incredibly long, stretching maybe a meter or more.
A meter for a single cell. That's wild.
It is. And shape varies just as much. Cells can be disc-like red blood cells, or polygonal, columnar, cuboid, thread-like. Some are completely irregular, changing shape.
And that shape relates to function.
Always. Form follows function in biology. The long, thin shape of a nerve cell is perfect for transmitting signals. The biconcave disc shape of a red blood cell maximizes surface area for oxygen exchange. It's all beautifully adapted.
Okay, let's zoom in on the prokaryotes first. The bacteria and their relatives.
Right. So besides bacteria, this group includes blue-green algae or cyanobacteria, mycoplasma, and PPLO, that stands for pleuropneumonia-like organisms. They're generally smaller, and crucially, they multiply much, much faster than eukaryotic cells.
What's their basic setup inside?
Pretty straightforward, relatively speaking. Most have a cell wall outside their plasma membrane. Mycoplasma are an exception. They don't have one. Inside, you've got the cytoplasm. Their genetic material is typically a single circular chromosome just kind of floating in a region called the nucleoid. No nuclear membrane.
You called it naked DNA earlier.
Yeah, exactly. Not enclosed. And here's something really interesting and important. Many bacteria also have extra bits of small circular DNA called plasmids.
Plasmids, what do they do?
They can carry extra genes, things not essential for basic survival, but maybe providing an advantage, like famously resistance to antibiotics. A bacterium can gain resistance by picking up a plasmid carrying a resistance gene. This is a huge issue in medicine, obviously.
Yeah, I can see that. So plasmids are key. What else is characteristic?
Well, they don't have most of the complex membrane-bound organelles you find in eukaryotes. No mitochondria, no ER, none of that. They do have ribosomes for making proteins, but they're slightly different, smaller. We call them 70S ribosomes. And they store reserve materials in simple inclusion bodies in the cytoplasm.
You also mentioned mesosomes before, unique folds.
Yes, mesosomes. These are characteristic infoldings of the plasma membrane in prokaryotes. They're not just random wrinkles. They seem to have specific roles.
Like,
They're involved in things like forming the cell wall during division, helping replicate and separate the DNA, respiration. Some enzymatic processes happen there. They also increase the membrane's surface area.
okay, and that outer boundary, the cell envelope, you said it's 3 layers.
Often, yes, it's a complex protective unit. On the very outside, you might have a glycocalyx that can be a loose slime layer or a tough capsule. Then comes the cell wall, providing rigidity, and finally the plasma membrane controlling what gets in and out. These layers act together.
And that relates to Gram staining.
Exactly. The differences in the cell envelope structure, particularly the cell wall thickness and composition, determine whether bacteria stain gram-positive, purple, or gram-negative, pink. It's a fundamental classification method, really useful for identifying bacteria.
What about movement? How do they get around?
Many bacteria have flagella, these long whip-like filaments that rotate and propel the cell. A bacterial flagellum has 3 parts: filament, hook, and basal body, which anchors it and acts as a motor.
Different from eukaryotic flagella you mentioned.
Structurally very different, yes, even though the function is similar. Prokaryotes might also have pili and fimbriae. These are shorter, hair-like structures on the surface, but they're generally used for attachment sticking to surfaces or to other cells, not really for motility like flagella.
Our ribosomes, you said 70S, so they just make proteins.
Yep, protein synthesis factories, just like in eukaryotes, but the 70S size is distinctive. They're made of 2 subunits. 50S and 30S, the numbers don't add up simply, it's about sedimentation rate. Often you'll see several ribosomes attached to a single messenger RNA molecule, forming what's called a polyribosome or polysome. It's like an assembly line, translating the mRNA code into multiple protein copies simultaneously. Very efficient.
And those inclusion bodies, just storage bins.
Pretty much. They're not membrane-bound, just granules or aggregates of reserve materials floating in the cytoplasm. Could be phosphate, glycogen, fats. Some aquatic prokaryotes even have gas vacuoles, which help them regulate buoyancy.
Okay, that gives us a good picture of prokaryotes. Now let's switch gears to the eukaryotes. Plants, animals, fungi, protists. What's the big picture there?
Complexity and compartmentalization, that's the key difference. Eukaryotic cells have their genetic material neatly packaged inside that membrane-bound nucleus. But beyond that, their cytoplasm is filled with various membrane-bound organelles, each performing specific tasks. It's like a city with different districts and factories.
Mitochondria, Golgi, all those.
Exactly. Plus, they have a complex cytoskeleton for structure and movement, and their DNA is organized into multiple linear chromosomes associated with proteins.
Are plant and animal cells basically the same, apart from the cell wall?
Mostly similar, but there are some really important distinctions. Plant cells, as we said, have that rigid cell wall outside the plasma membrane. They also have plastids, chloroplasts for photosynthesis being the most famous, and typically a very large central vacuole.
And animal cells lack those.
Generally, yes. Animal cells lack cell walls, plastids, and that large central vacuole. But animal cells usually have centrioles, which are involved in cell division, and those are typically absent in higher plants. So definite differences reflecting their different lifestyles.
Okay, let's dive into some of those key eukaryotic components. The cell membrane first. The boundary.
Right, the plasma membrane. Its structure wasn't really clear until the electron microscope came along in the 1950s. We now know it's mainly lipids and proteins.
What kind of lipids?
Primarily phospholipids. They have that special structure, a hydrophilic, water-loving head, and two hydrophobic, water-fearing tails. In water, they naturally arrange themselves into a bilayer, 2 layers thick, with the tails pointing inwards, away from the water, and the heads facing outwards. There's also cholesterol tucked in there, especially in animal cells, which affects how fluid the membrane is.
And protein.
They're embedded within or attached to this lipid bilayer. Some go all the way through integral proteins, while others are just on the surface peripheral proteins. These proteins do a lot of the membrane's work. Transport, acting as receptors, enzymes.
And this relates to the fluid mosaic model.
Precisely. Singer and Nicholson proposed this model in 1972, and it's still our best description. It views the membrane as a mosaic of proteins floating or drifting laterally within a fluid lipid bilayer. It's not rigid, it's dynamic.
Why is that fluidity important?
Oh, it's crucial for many things. Cell growth, forming junctions between cells, secretion of substances, endocytosis, taking things in, cell division. All these processes require the membrane components to be able to move and rearrange.
And it's a selective barrier too, right? Controls traffic.
Absolutely key. It's selectively permeable. Some things can cross easily, others can't. Small, uncharged molecules might just diffuse across. Following their concentration gradient, that's passive transport, requires no energy. Water moves by a specific type of diffusion called osmosis.
But what about bigger things or charged things?
They usually need help, often from those membrane proteins. If it's still moving down the concentration gradient, it might be facilitated diffusion, still passive. But if the cell needs to move something against its concentration gradient...
Like pumping something uphill.
Exactly. That requires energy, usually from ATP. That's active transport. The sodium-potassium pump is a classic example. Constantly pumping sodium out and potassium into our cells against their respective gradients. Vital for nerve function and much more.
Okay, membrane covered. What about the cell wall in plants and fungi?
Right, that's the layer outside the plasma membrane in those organisms. It's non-living, pretty rigid, and gives the cell its definite shape.
What's its job besides shape?
Several things. It protects the cell from mechanical stress and infection. It helps prevent excessive water uptake that could burst the cell. It also plays a role in cell-to-cell interactions, but it's generally permeable, not a major barrier to small molecules like the plasma membrane is.
Does the composition vary?
Yes. In algae, it might be cellulose, galactans, mannans. In other plants, it's primarily cellulose. Fungi use titin. A young plant cell has a flexible primary wall, allowing growth. Later, a thicker, more rigid secondary wall often forms inside the primary wall.
And how do adjacent plant cells connect?
They're cemented together by a layer called the middle lamella, which is rich in calcium pictate, acts like glue, but they're not totally isolated. There are fine cytoplasmic strands called plasmodesmata that pass through the cell walls and middle lamella, directly connecting the cytoplasm of neighboring cells. Allows for communication and transport between them.
Okay, now for this endomembrane system, you said it's a coordinated team.
That's a good way to put it, yeah. It's a group of organelles in eukaryotic cells whose membranes and functions are interconnected either directly or indirectly through vesicle transport. This system includes the endoplasmic reticulum, ER, the Golgi apparatus, or Golgi complex, lysosomes, and vacuoles.
And you specifically said mitochondria and chloroplasts are not part of it.
Correct. Also peroxisomes, their functions aren't directly coordinated with that flow of membrane and materials through the ER, Golgi, etc. They're kind of independent players in that sense.
All right, let's break down the team members. Start with the ER, endoplasmic reticulum.
Right. The ER is this extensive network of tiny tubules and flattened sacs called cisternae spreading throughout the cytoplasm. It basically divides the intracellular space into two compartments: the luminal space, inside the ER membranes, and the extraluminal space, the cytoplasm proper.
And there are two types, rough and smooth.
Yes. Rough ER, or ER, looks rough under the electron microscope because it's studded with ribosomes on its outer surface.
Ribosomes making proteins.
Exactly. So RER is heavily involved in synthesizing proteins that are destined for secretion out of the cell, or insertion into membranes, or delivery to other organelles like the Golgi or lysosomes. It's often continuous with the outer membrane of the nuclear envelope.
And smooth ER.
Smooth ER, SER, lacks ribosomes, hence smooth. It has different functions. It's a major site for lipid synthesis, including steroids like hormones. It's also involved in detoxification processes in some cells, like liver cells, and in storing calcium ions.
Okay, from the ER, things often go to the Golgi apparatus.
That's the typical flow, yes. The Golgi apparatus, named after Camillo Golgi, who first saw it in 1898, is like the cell's post office, or processing and packaging center. It consists of a stack of flattened, membrane-bound sacs called cisternae.
Stacks of sacs. Does it have a direction?
It does. The stack has a distinct polarity. There's a cis face, or forming face, which is usually oriented towards the ER. This is where materials arrive from the ER, typically in small membrane bubbles called transport vesicles. Then there's a trans face, or maturing face, on the opposite side, where processed materials leave in new vesicles.
What happens inside the Golgi?
It receives proteins and lipids from the ER, modifies them further, like adding carbohydrate chains to form glycoproteins or glycolipids, sorts them, and then packages them into vesicles for delivery to their final destination. Could be lysosomes, the plasma membrane for secretion, or other locations.
Okay, so processing and shipping. What about lysosomes? You said they're part of this system too.
Yes, lysosomes are essentially small vesicles that bud off from the Golgi apparatus. They're the cell's recycling and digestion centers.
Recycling how?
They contain a powerful cocktail of hydrolytic enzymes, lipases, proteases, carbohydrates, nucleuses that can break down all sorts of macromolecules. Worn-out organelles, material taken into the cell by endocytosis, debris. These enzymes work best at an acidic pH, which is maintained inside the lysosome.
So they break things down safely inside their own compartment.
Precisely. It compartmentalizes the digestion.
And the last member of the system, vacuoles.
Right. Vacuoles are membrane-bound sacs, but their roles can be quite varied. The membrane surrounding a vacuole is called the tonoplast. In plant cells, there's usually one very large central vacuole that can take up a huge percentage of the cell volume, up to 90%.
What's in that huge plant vacuole?
Mainly water, but also ions, nutrients, pigments, waste products. It plays a crucial role in maintaining pervert pressure, keeping the plant cell firm. The tonoplast actively pumps ions into the vacuole, which helps draw water in.
Do animal cells have vacuoles?
They can, but they're usually much smaller and more temporary than the large central vacuole in plants. Think maybe small vacuoles for storage or transport. Some specialized vacuoles exist too, like contractile vacuoles in freshwater protists like amoeba, which pump out excess water. And food vacuoles form when cells engulf food particles.
Okay, that covers the endomembrane system. Let's move to the energy specialists.
Yeah.
Mitochondria, the powerhouses.
That's the classic nickname, and it's accurate. Mitochondria are where the main energy-releasing process of aerobic respiration happens. They generate most of the cell's supply of ATP, the main energy currency.
What do they look like?
Typically sausage-shaped or cylindrical, though they can vary. The key feature is their double membrane. There's a smooth outer membrane and then a highly folded inner membrane.
Those folds have a name, right?
Cristae. Yes, these folds, the cristae, dramatically increase the surface area of the inner membrane. And that's important because many of the enzymes and protein complexes involved in ATP synthesis are embedded in that inner membrane.
And inside the inner membrane
is the mitochondrial matrix. This space contains enzymes for other stages of respiration, and also, this is fascinating, mitochondria have their own DNA. It's usually a single circular molecule, much like bacterial DNA. They also have their own ribosomobins, 70S, like bacteria in RNA.
Their own DNA and ribosomes, so they can make some of their own proteins.
They can. And they reproduce by dividing. through a process called fission, similar to bacterial division. This is strong evidence for the endosymbiotic theory, the idea that mitochondria originated from ancient aerobic bacteria that were engulfed by an early eukaryotic cell and formed a mutually beneficial relationship.
Wow, ancient bacteria living inside our cells. Okay, what about the other big energy-related organelles, plastids, found in plants?
Plastids, yes. Found in all plant cells and also in euglenoids. They're also typically large and often easily visible under a light microscope because they bear pigments. Based on the pigment type, we classify them.
What are the types?
3 main ones: chloroplasts, chromoplasts, and leucoplasts.
Chloroplasts are the green ones.
Yeah.
Photosynthesis.
Exactly. They contain chlorophyll, the green pigment that captures light energy, as well as accessory carotenoid pigments. This is where photosynthesis takes place, converting light energy into chemical energy stored in sugars.
Okay, chromoplasts.
Chromoplasts contain other types of fat-soluble pigments, mainly carotenoids like carotene orange and xanthophylls, yellow. They give the yellow, orange, or red colors to many fruits, flowers, and roots like carrots or tomatoes.
And leucoplasts.
These are colorless plastids. Their main job is storage. Depending on what they store, they have different names. Amyloplasts store starch, ileoplasts store oils and fats, and aleuroplasts store proteins.
What's the structure of a chloroplast like? Similar to a mitochondrium.
Similar in having a double membrane, yes. An outer and an inner membrane. The space inside the inner membrane is called the stroma. The stroma contains enzymes for the sugar-building reactions of photosynthesis, the Calvin cycle. And just like mitochondria, chloroplasts have their own circular DNA and 70S ribosomes.
So chloroplasts are also thought to be endosymbionts from photosynthetic bacteria.
That's the leading theory, yes. Within the stroma, there is a third membrane system, flattened interconnected sacs called thylakoids. The chlorophyll pigments are located within the thylakoid membranes. In many chloroplasts, thylakoids are stacked up like coins into structures called grana, singular, granum.
Okay, so mitochondria for energy release, chloroplasts for energy capture. Now, what about the basic machinery, like ribosomes?
Ribosomes, yeah. Crucial little things. George Palade first observed them as dense particles back in 1953. They're made of ribosomal RNA, rRNA, and proteins. And a key point, they are not surrounded by any membrane.
No membrane, unlike most other organelles.
Correct. They're just complexes of RNA and protein, and they come in slightly different sizes. Eukaryotes have the larger 80S ribosomes found free in the cytoplasm and also attached to the RER. Prokaryotes, as we said, have smaller 70S ribosomes. Chloroplasts and mitochondria also have 70S ribosomes.
What does the S mean again?
Svedberg unit. It's a measure of sedimentation rate in a centrifuge, basically related to size and shape. So an 80S ribosome have a 60S large subunit and a 40S small subunit. A 70S has a 50S and a 30S subunit. The numbers aren't additive.
And their job is always protein synthesis.
Always. They are the site where the genetic information encoded in messenger RNA, mRNA, is translated into a sequence of amino acids, building a protein chain. Whether free or attached, that's their fundamental role.
Now, what holds the cell's shape and allows movement? The cytoskeleton.
Exactly. The cytoskeleton is this intricate network of protein filaments and tubules extending throughout the cytoplasm of eukaryotic cells. It's not static, it's dynamic, constantly assembling and disassembling.
What's it made of?
3 main types of protein structures: microtubules, hollow tubes made of tubulin; microfilaments, thin solid rods often made of actin; and intermediate filaments, rope-like fibers made of various proteins.
And its functions.
Many. It provides mechanical support and maintains cell shape. It anchors organelles. It's involved in cell movement, both the movement of the entire cell, like an amoeba crawling, and the movement of things within the cell, like vesicles traveling along microtubule tracts. It's also crucial for chromosome separation during cell division.
Okay, structural support and tracks. What about cilia and flagella? The hairs and tails.
Right. These are hair-like outgrowths from the cell membrane used for movement. Cilia are usually short and numerous, and they beat in a coordinated way, like tiny oars, to either move the cell or move fluid over the surface of the cell, like in our respiratory tract.
And flagella.
Flagella are typically much longer and usually fewer in number, often just 1 or 2 per cell. They generate movement with a more whip-like, undulating motion. Think of a sperm cell's tail.
And eukaryotic flagella are different from prokaryotic ones.
Structurally very different, yes. Prokaryotic flagella rotate like propellers. Eukaryotic flagella bend and flex. Internally, eukaryotic cilia and flagella have a characteristic structure. Their core is called the axonome.
The axonome. What's that like?
It typically consists of 9 pairs, doublets, of microtubules arranged in a ring around 2 single microtubules in the center. This is known as the 9+2 array. It's a remarkably conserved structure across diverse eukaryotes. These microtubules slide past each other, powered by motor proteins, causing the cilium or flagellum to bend, and they emerge from a structure at their base called the basal body.
Basal body. Sounds like centrioles.
Very similar structure,
Yeah.
which brings us to the centrosome and centrioles. The centrosome is the main microtubule organizing center in animal cells. It usually contains 2 centrioles, positioned perpendicular to each other.
What are centrioles made of?
Each centriole is a cylinder made of 9 triplets of microtubules arranged in a cartwheel pattern. No central microtubule, so it's a 9+0 arrangement of triplets. They're surrounded by amorphous pericentriolar material, which is actually where most microtubule nucleation starts.
What do they do?
They are involved in organizing the mitotic spindle during cell division in animal cells, helping to pull the chromosomes apart. And as you noted, centrioles also form the basal bodies from which cilia and flagella grow.
Okay, almost there. The big boss. The nucleus. The control center.
The control center, indeed. First described way back by Robert Brown in 1831. Later, Fleming coined the term chromatin for the material inside.
What does the nucleus look like when the cell isn't dividing? The interphase nucleus.
During interphase, the genetic material exists as chromatin, this diffuse, extended complex of DNA, tightly wound around proteins, mainly histones. It looks kind of like a tangled mass under the microscope. The nucleus also contains the nuclear matrix, sort of internal framework, and 1 or more dense regions called nucleoli, singular,
Nucleolus.
and it's enclosed by that envelope.
Yes, the nuclear envelope. A double membrane, an inner and an outer membrane, separated by a space called the paranuclear space. The outer membrane is actually continuous with the endoplasmic reticulum and often has ribosomes attached.
How do things get in and out? It's
Yes.
a barrier, right? It is, but it's perforated by nuclear pores. These aren't just simple holes. They're complex structures formed where the inner and outer membranes fuse. They act as regulated gateways, controlling the passage of molecules, especially proteins going into the nucleus and RNA coming out.
Do all eukaryotic cells have a nucleus?
Most do. Typically 1 per cell, but there are exceptions. Some cells are multinucleated, and some mature cells actually lose their nucleus entirely. Good examples are mammalian red blood cells and the sieve tube cells in plants that transport sugars.
Wait, they lose their nucleus. How are they still living? That's a fascinating question.
It is. They're metabolically active for their lifespan, carrying out their specific function, like oxygen transport, but they can't divide or synthesize proteins anymore. They rely on proteins made earlier or provided by neighboring cells. It raises interesting points about what defines living at the cellular level.
Definitely something to think about. What about the nucleolus inside the nucleus?
The nucleolus is that dense structure within the nuclear plasm, the cytoplasm of the nucleus. It's not membrane-bound. Its main job is ribosome synthesis. It's where ribosomal RNA, rRNA, is transcribed and combined with proteins imported from the cytoplasm to form the ribosomal subunits.
So busy cells making lots of protein would have prominent nucleoli.
Exactly. Cells with high rates of protein synthesis usually have large or multiple nucleoli.
Okay, back to chromatin. That's the DNA plus proteins.
Correct. DNA carries the genetic code, and proteins, especially histones, help package and condense that incredibly long DNA molecule so it fits inside the nucleus. Think about it. A single human cell has about 2 meters of DNA.
2 meters in that tiny nucleus? How?
Through incredibly efficient packaging. The DNA wraps around histone proteins to form structures called nucleosomes, like beads on a string, which then coil and fold further and further into the compact structure we call chromatin.
And that chromatin condenses even more to form visible chromosomes during cell division.
Precisely. When the cell prepares to divide, the chromatin fibers condense dramatically, becoming visible as distinct chromosomes under the light microscope. Each duplicated chromosome consists of 2 identical sister chromatids joined together at a constricted region called the centromere.
Centromere. The waist.
Kind of, yeah. And on the surface of the centromere are protein structures called kinetochores. These are what the spindle microtubules attach to during cell division to pull the sister chromatids apart.
Do all chromosomes look the same?
No, their appearance depends largely on the position of the centromere. If it's right in the middle, it's metacentric. Slightly off-center, submetacentric. Near one end, acrocentric. Right at the tip, telecentric. This morphology is characteristic for each chromosome pair. Some chromosomes might also have secondary constrictions or small fragments called satellites at their ends.
Okay, that covers the nucleus. One last thing mentioned in the outline, microbodies.
Ah, yes. Microbodies are just a general term for small, membrane-bound vesicles found in the cytoplasm of both plant and animal cells. They contain various enzymes and are involved in specific metabolic pathways. Peroxisomes, for example, are microbodies involved in breaking down fatty acids and detoxifying harmful substances.
Got it. So that's quite a tour.
It really is. And when you step back, despite all that complexity and the differences between, say, a bacterium and a human neuron, the amazing thing is the underlying unity. The cell truly is the fundamental unit. That cellular organization is the common thread running through all known life.
And understanding all these parts, how they work, how they interact, that reductionist approach we mentioned, that's how we really grasp life's processes, right? Normal function, and also when things go wrong in disease.
Absolutely. Understanding the molecular machinery allows us to understand physiology and pathology at their deepest levels. It's the foundation for so much of modern medicine and biotechnology. It bridges that gap between the microscopic world and our everyday health.
So, reflecting on all this incredible complexity packed into something so tiny, it makes you wonder, doesn't it? Given how much intricate machinery is working inside every single cell, what astonishing discoveries are still waiting for us? What new levels of understanding about health, disease, maybe even the very nature of life are hidden in those molecular details?
It's a frontier that keeps expanding. There's always more to learn, more intricate mechanisms to uncover. So yes, keep marveling at that unseen world inside us. It's where the secrets are.